Dear friends, good morning. Today we can discuss about enzymology, enzyme classification, nomenclature and IUB system of enzyme classification. Nomenclature of enzyme classification. When enzymes were first discovered, they were given various unsystematic names by their discoverers, such as pepsin, trypsin, ptyalin, and also zymase. Later on, enzymes were named by adding suffix asc to the name of substrate. For example, any substrates involving the enzymatic reaction, the name. Example, esterase which acts on ester, amylase which acts on starch, urease which acts on urea. Furthermore, the name of some enzymes also denotes the type of reaction along with substrate. Example for lactate dehydrogenase enzyme. This enzyme catalyzes the dehydrogenation reaction, removal of hydrogen of lactic acid. However, some enzymes were named before these general rules were formulated, and such enzymes have retained their trivial names. Example, pepsin, trypsin. There is no perfect method for classification of enzymes. However, the most comprehensive system for the classification of enzymes was devised in 1964 by the Enzyme Commission of the International Union of Biochemistry and Molecular Biology, shortly called as IUBMB. The major features of the IUBMB system for classification of enzymes. The enzymes are classified first according to the general type of chemical reaction they catalyze. On this basis, there are 6 important classes. Each class is further classified into several subclasses on the basis of the type of bond split or formed, chemical group removed or transferred, or some case by a simple subclassification of general reaction type. Enzyme is given a system code number commonly known as Enzyme Commission number, EC number. Example 1.1.1.1. This is Enzyme Commission for alcohol dehydrogenases. Here first digits indicates the characterization of reaction type. Second digits indicates the subclasses of enzyme. Third digits subclasses. Fourth digit indicates the particular enzyme named. Although the IUB system is complex in the precise, descriptive, informative one. These are the 6 major classes of enzyme. Class 1, oxidoreductase, which catalyzes the oxidation reduction reaction. Second one class, transferases. This enzyme catalyzes the transfer of functional groups. Third one is hydrolases. This enzyme catalyzes the hydrolysis reaction. Fourth one, lyases. This enzyme catalyzes the group elimination to form the double bond. Fifth one, isomerases, which involves the isomerization reactions. Sixth one is ligases. This enzyme involved in joining enzyme. Here new bond will be formed with the help of ATP hydrolysis. First you will see about class 1, oxidoreductase. This group of enzyme will catalyze oxidation of one substrate with simultaneously reduction of another substrate or coenzyme, or transfer of hydrogen or addition of oxygen. Here shortly represented as A minus plus B gives A plus B minus. Example, alcohol reacts with NAD plus in the presence of alcohol dehydrogenase enzyme, which gives the aldehyde plus NADH H plus. Reduced form. NAD will be reduced to NADH H plus. The enzyme is alcohol dehydrogenase. IUB name is alcohol NAD oxidoreductase. Enzyme commission number of alcohol dehydrogenase 1.1.1.1. Oxidoreductases may also oxidize substrates by adding oxygen. This is for diagrammatic representation of oxidoreductase reaction. A substrate reduced form, B substrate oxidized form. During the oxidoreductase reaction, A converted as a oxidized form, B converted as reduced form. Here some more enzymes also under the category of oxidoreductases. Example for dehydrogenases, oxidases, peroxidases, reductases, monooxygenases, dioxygenases. These are the under the category of oxidoreductase enzyme. Next move on to second class, transferases. This class of enzyme transfers one group from one substrate to another. This may be represented as AR plus B gives A plus BR. Here R as a one group. For example, hexose plus ATP which gives the hexose 6-phosphate plus ADP, adenosine diphosphate. The name of enzyme is hexokinase and systematic name is ATP hexose 6-phosphotransferase. This is for diagrammatic representation of transferases reaction. Here first one, AB reacts with C which gives the A alone plus BC. Here AB having some B part transferred to C. Some more examples for transferases enzyme, transferases, glycosyl transferases, aminotransferases, phosphotransferases. These are the corresponding group will be transferred. Third one is class 3, hydrolysis. This class of enzyme can hydrolyze esters, ether, peptide or glycosidic bonds by adding water then breaking the bond. General reaction, AB reacts with water to gives the AH plus BOH. Water will be converted as H plus OH. Example, acetylcholine reacts with water to produce choline plus acetate. The enzyme is acetylcholine esterase or acetylcholine hydrolase. Systematic name of this enzyme, all digestive enzymes are hydrolysis. Here the diagrammatic representation of hydrolysis. First AB reacts with water which gives the AH plus BOH. Examples for hydrolysis enzyme, esterases, glycosidases, peptidases, amidases. Next move on to class 4, lyases. These enzymes can remove groups from substrate or break the bonds by mechanisms other than hydrolysis. Example, fructose 1,6-bisphosphate converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Here aldolase as a enzyme which converts the fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and also dihydroxyacetone phosphate. This is the diagrammatic representation, A plus B which gives the AB. Here bonds will be breaked. For example, CC lyases, CO lyases, CN lyases or CS lyases. These bonds will be breakdown. Next one, class 5, isomerases. These enzymes can produce optical, geometrical or positional isomers of substrates. Example, racenases, epimerases, cis-trans isomerases are the examples of isomeric enzyme. Example for isomeric enzyme, glyceraldehyde 3-phosphate which converted as dihydroxyacetone phosphate in the presence of triose phosphate isomerase enzyme. Here diagrammatic representation, A, some groups transferred to another position, iso A. Example for isomerases, epimerases, cis-trans isomerases, intramolecular transferases. Next one, group 6 or class 6 ligases. These enzymes which involve the joining purpose, these enzymes catalyze the synthesis of new bonds coupling with the breakdown of ATP or other nucleoside biphosphate. For example, acetyl-CoA plus carbon dioxide in the presence of ATP which converts malanyl-CoA plus ADP plus inorganic phosphate. The catalyzed enzymes are acetyl-CoA carboxylase. Below, A plus B in the presence of XTP. Here X as a nucleotide basis either adenine or guanine or uracil or cytosine. N1 nucleotide basis or nucleotides reacts with molecule to produces the diphosphate form, ADP or UTP or CTP or GTP. In this way, triphosphate converted as diphosphate form. Here some examples: CC ligases, CO ligases, CN ligases and CS ligases. These are the enzyme classification of